Sickle cell disease, SCD, is a severe monogenic disorder characterized by the production of abnormal hemoglobin S, HbS, leading to erythrocyte rigidity, vaso-occlusion, and chronic multi-organ complications. While hematopoietic stem cell transplantation, HSCT, and gene therapy via ex vivo editing have emerged as transformative strategies, the advent of in vivo gene therapy presents a new frontier for scalable, minimally invasive, and potentially curative treatment approaches. In vivo gene therapy leverages molecular tools such as mRNA-based gene editing, CRISPR-Cas9, base editing, and prime editing to correct or modulate genetic aberrations directly within the body. This paper provides a deep dive into the mechanisms, clinical advancements, challenges, and future perspectives of mRNA-based in vivo gene therapy for sickle cell disease, highlighting actionable therapeutic targets and translational potential. In vivo gene therapy utilizes targeted delivery of genetic material to modify gene expression within an individual's own cells. Unlike ex vivo approaches which require cell extraction and reinfusion, in vivo strategies introduce genetic modifications directly into the patient's body through various delivery systems, including lipid nanoparticles, LNPs, utilized for delivering mRNA therapeutics efficiently to hematopoietic stem and progenitor cells, HSPCs, in vivo. Adeno-associated viruses, AAVs, engineered vectors for precise gene delivery, often optimized for hematopoietic targeting. Non-viral methods include nanoparticles, electroporation, and peptide-based carriers that enhance safety by avoiding immune responses associated with viral vectors. Key mechanisms of action include mRNA-based gene therapy involves encoding functional proteins or genome editing machinery to correct the SCD mutation. Then, there's base editing and prime editing, which repair the pathogenic HBB gene without creating double-stranded breaks. Regulation of fetal hemoglobin, or HbF, expression involves targeting transcription factors such as BCL11A to induce HbF production, thereby mitigating the pathophysiology of SCD. While in vivo gene therapy offers groundbreaking potential, it comes with both risks and benefits. Benefits include being minimally invasive, with no need for myeloablative conditioning or stem cell transplantation. It is potentially curative, as direct correction of pathogenic mutations in hematopoietic stem cells may lead to lifelong disease modification. Additionally, it offers scalability, making it easier to deploy in low-resource settings compared to ex vivo therapies. Real-world application of in vivo gene therapy for sickle cell disease is promising. Several preclinical and early clinical studies have explored mRNA-based and gene editing approaches for in vivo SCD therapy. mRNA therapeutics for fetal hemoglobin induction are particularly noteworthy. Several biotech firms are developing mRNA-based reprogramming of hematopoietic stem and progenitor cells to induce gamma globin expression, promoting fetal hemoglobin production to counteract sickling. CRISPR/Cas9 in vivo editing is another exciting development. Advances in targeted AAV CRISPR delivery are demonstrating feasibility in permanently correcting the HBB mutation in murine models. Emerging studies also focus on prime and base editing clinical research. These studies are investigating adenine base editing, known as ABE, and prime editing to repair the pathogenic sickle mutation at the DNA level without triggering unwanted cellular responses. While in vivo gene therapy presents a scalable solution for SCD, key challenges include efficient hematopoietic cell targeting. Current nanoparticle and viral delivery systems require optimization to ensure precise in vivo HSPC targeting. Manufacturing and cost constraints. Large-scale production of LNPs and AAV vectors remains a limiting factor. Regulatory hurdles. Long-term safety data is needed to satisfy regulatory bodies before widespread clinical adoption. Ethical considerations. Ethical concerns surrounding in vivo gene therapy include germline editing risks, ensuring somatic cell specificity to prevent unintended heritable modifications, equitable access, preventing a disparity between high-resource and low-resource regions in accessing gene therapy, Informed consent, particularly for pediatric applications where long-term risks are uncertain. Ongoing clinical trials and research in mRNA-based gene therapy for SCD. Recent clinical advancements include Vertex Pharmaceuticals and CRISPR Therapeutics, investigating in vivo CRISPR-based strategies for hemoglobinopathies. Moderna and Pfizer's mRNA platforms, exploring LNP-delivered mRNA therapeutics for hematopoietic disorders. NIH/NHLBI-sponsored trials, examining direct in vivo reprogramming of HSPCs to enhance HbF levels. Future outlook. In vivo gene therapy is poised to revolutionize SCD treatment, offering a minimally invasive, potentially curative alternative to current interventions. Key areas of future research include next-generation nanoparticle engineering, improving HSPC targeting efficiency for safer mRNA delivery. Expanded clinical trials establishing safety and efficacy profiles for mRNA-based in vivo editing, global implementation strategies, developing cost-effective models to bring in vivo gene therapy to resource-limited settings. Conclusion. The landscape of mRNA-based in vivo gene therapy for sickle cell disease is rapidly evolving, presenting a transformative opportunity to cure SCD at the molecular level. Overcoming current barriers in delivery mechanisms, ethical frameworks, and global accessibility will be critical to ensuring that these cutting-edge therapies translate from bench to bedside, improving the lives of millions affected by SCD worldwide.